Hello friends, my name is Mr. Desley SD. Work with Mahatma Gandhi Vidya Mandirs, Janta Vidyalay and Junior College Manur, Taluka Kalwan, District Nasik. My subject is biology, and today we are going to study now chapter number 4, molecular basis of inheritance. First topic in this chapter, the discovery of DNA. The modern understanding of DNA has evolved from the discovery of nucleic acid to the development of the double helix model in 1869, and its mature began works with white blood cells, which are the major component of pus from infections. He collected a lot of pus from the bandages at the local hospitals. He used a salt solution to wash the pus of the bandages. When he added a weak alkaline solution to the cells lysed and nuclei precipitated out of the solutions. From the cell's nuclei, he isolated a unique chemical substance to which he called nuclein. Chemically, nuclein has high phosphorus content. Moreover, he showed acidic properties, hence, it was named as nucleic acid. By the early 1900s, we knew that nuclein was a mix of proteins and nucleic acids. There are two kinds of nucleic acids, DNA and RNA. Next topic is the genetic material is a DNA. By the early 1900s, geneticists knew that gene control the inheritance of trait, that genes are located on chromosomes, and that chemically chromosomes are mainly composed of DNA and proteins. Initially, most geneticists thought that proteins are large, complex molecules and store information needed to govern cell metabolism. Hence, it was assumed that proteins cause the variations observed within species. On the other hand, DNA thought to be small, simple molecule whose composition varied little among species. Over the time, these ideas about DNA were shown to wrong. In fact, DNA molecules are large and vary tremendously within the and among species. Variation in the DNA molecules are different than the variations in shape, electrical charges, and function shown by proteins. So, it is not surprising that most researchers initially favored protein as the genetic material. Over a period of roughly 25 years, geneticists became convinced that DNA and not protein was the genetic material. Let us study three important contributions that help cause the shift of opinions. Next topic, that is Griffith's experiment. In 1928, a British medical officer, Frederick Griffith, performed an experiment on bacterium Streptococcus pneumoniae that causes pneumonia in humans and other mammals. Griffith used two strains or two genetic varieties of Streptococcus to find a cure for pneumonia, which was a common causes of death at that time. The two strains used were number one, virulent, smooth, pathogenic, and encapsulated S type. Second one, non-virulent, rough, non-pathogenic, and non-capsulated Rs type. Griffith conducted 4 experiments on this bacteria. First one is he ejected bacteria of strain R to mice. The mice survived because it did not develop pneumonia. Second, when he injected bacteria of strain S to mice, the mice developed pneumonia and died. Third experiment, he injected hit-kill strain S bacteria to mice. Once again, the mice survived. And fourth experiment, he mixed hit-killed S bacteria with live bacteria of strain R and injected to mice. The mice died and Griffith recovered large number of live strain S bacteria from the blood of the dead cells. Griffith concluded that the R strain bacterium must have taken up to what he called a transforming principles from the hit-killed S bacterium, which allowed R strain to get transformed into smooth coated bacterium and become virulent as shown as the figures. Next topic, next experiment, that is Avery, McCarthy and MacLeod experiment. In 1944, after some 10 years of research and this experimentation, all these scientists first evidence to prove the DNA is genetic material. Through the experiment, they purified DNA, RNA, proteins, enzymes, and other material from cell free extracts of S cells strains and mixed with heat killed S strain and R cells separately to confirm which one could transform living R cell into S cell. Only DNA was able to transform harmless strain R into deadly strain S. They also discovered that protein digesting enzymes, RNA digesting enzymes did not affect transformation. The transforming substance was neither a protein nor a RNA, and DNA digest with DNAs did inhibit the transformation, suggesting that DNA caused the transformation. This experiment proved that the transforming principle is DNA, but all biologists were not convinced. They shown as the figure. Finally, Hershey and Chase proved that DNA is the genetic material and not proteins by using bacteriophage. Next topic, next experiment, Hershey-Chase experiment. They work with virus that infect bacteria, that is bacteriophage, which are composed of DNA and protein. They used radioactive phosphorus in the medium for some viruses and radioactive sulfur for some others. They grew some viruses on the medium that contain radioactive phosphorus and some others on medium that contain radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, because DNA contain phosphorus but protein do not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Radioactive phages were allowed to infect E. coli bacteria grown on the medium containing normal phosphorus and sulfur. Then as the infection proceed. In other words, Sometime after infection, radioactivity for phosphorus and sulfur was tested. Only radioactive phosphorus was found inside the bacterial cell, indicating that DNA is the genetic material. And those scientists, with the help of diagram, they proved their experiment. Next topic, DNA packaging. Length of DNA double helix molecule in a typical mammalian cell is approximately 2.2 meters. Approximate size of typical molecule is 10 raised to 6 meter. How these long DNA molecules can be then accommodated in such a small nucleus, it therefore must be condensed, coiled and supercoiled to fit inside such small nucleus. DNA packaging consists of two types. Number one, packaging in prokaryotes, and number second, packaging in eukaryotes. Number one, packaging in prokaryotes. In prokaryotes like E. coli, cell size is almost 2.2 to 3 micron long. They do not have cell organized nucleus. It is without nuclear membrane and nucleolus. The nucleoid is small, circular, highly folded, naked ring of DNA, which is 1100 micron long in perimeter, containing about 4.6 million base pair. Nucleoid is to be fitted or packed into the cell, which is hardly 2 to 3 micron long. Hence, the negatively charged DNA becomes circular, reducing the size to 350 numerical meter in diameter. This is further reduced to 30 micron meter in diameter because of holding 40 to 50 domains or loops are formed. Formation of loops is assisted by RNA connector. Each domain is further coiled and supercoiled, thereby reducing the size down to 2 numerical in diameter. This coiled is assisted by positively charged protein and enzymes like DNA gyrase and DNA topoisomerase for the maintaining super. Second one is packaging in eukaryotes. Eukaryote shows well organized nucleus containing nuclear membrane, nucleolus and thread-like material in the form of chromosomes. In the chromosomes, DNA is associated with histone and non-histone proteins, as was reported by R. Kornberg in 1974. The organization of DNA is much more complex in eukaryotes, depending upon the abundance of amino acid residue with charge side chains. A protein acquired its charge. Histone are the proteins that are rich in lysine and adenine residue. Both these amino acid residue are basic amino acids and carry positive charges with them. So histones are a set of positively charged basic proteins. These histones organize themselves to make a unit of 8 molecules known as histone octamers. Next topic, that is DNA replication. It is most important topic in the chapter molecular basis of inheritance. DNA replication shows the DNA molecules regulate the control all the activities of the cell. Because of its unique structure, it is able to control the synthesis of other molecules of the cell. At the same time, when the cell reproduces, the DNA also should duplicate itself to distribute equally to the daughter cell. As a carrier of genetic information, DNA has to perform two important functions. First one is heterocatalytic function and second one is autocatalytic function. Heterocatalytic function, when DNA directs the synthesis of chemical molecules other than itself, then such functions of DNA are called as heterocatalytic function. Example, synthesis of RNA and synthesis of protein. Second one, autocatalytic function, when DNA directs the synthesis of DNA itself, when such function of DNA is called as autocatalytic function. Example is replication. The process by which DNA duplicates itself is called as replication. Through replication, it forms two copies that are identical to it. In eukaryotic organism, replication of DNA takes place only once in the cell cycle. It occurs in the S phase of interphase in the cell cycle. DNA replicates through semi-conservative mode of replication as shown in the figure. In eukaryotic organism, replication of DNA takes place only once in the cell cycle. It occurs in the S phase of interphase in the cell cycle. DNA replicates through semi-conservative mode of replication, and this replication is shown as below. Number 1, activation of nucleotides. The 4 types of nucleotides of DNA that is present in the nucleoplasm, they are activated by ATP in the presence of an enzymes phosphorylase. This results in the formation of deoxyribonucleotides, triphosphates. This process is known as phosphorylation. Number second step, point of origin or initiation point. It begins at the specific point origin and terminates at the point T. Origin is flanked by T site. The unit of DNA in which replication occurs is called as replicon. In prokaryotes, there is only one replicon. However, in eukaryotes, there are several replicons in tandem. At the point of origin, enzyme endonuclease nicks one of the strand of DNA temporarily. The nick occurs in the sugar phosphate backbone or phosphodiester bond. Number 3, third step, unwinding of DNA molecules. Now, enzymes DNA helicase operates by the breaking weak hydrogen bond in the vicinity of origin or O. The strand of DNA separate and unwind. This unwinding of bidirectional and continues as Y-shaped replication fork. Each separated strand acts as template. The two separated strands are prevented from recoiling. Single strand binding protein, SSB protein, remain attached to both the separated strands so as to facilitate synthesis of few polynucleotide chain or strands. Number 4, replicating fork. The point formed due to the unwinding and separation of two strands appear like Y-shaped fork called as replication fork. The unwinding of strand imposes strain which is relieved by superhelix relaxing enzymes. 5th point, synthesis of new strand. Each separated strand acts as mold or template for the synthesis of new complementary strand. It begins with the help of small RNA molecules called as RNA primer. RNA primer get associated with the third end of template strand and attract complementary nucleotides from surrounding nucleoplasm. These nucleotides molecule bind to the complementary nucleotides on the template strand by forming hydrogen bond. The newly bound nucleotides get interconnected by phosphoester bond, forming a polynucleotide strand. The synthesis of new complementary strand is catalyzed by the enzyme DNA polymerase. The new complementary strand is always formed in 5th to 3rd carbon direction. 6th step, leading and lagging strand. The template strand with free third end is called leading template and with free fifth carbon end is called lagging template. The process of replication is always at start at carbon 3 end of template strand and proceed towards carbon fifth end, as both the strands of the parental DNA are antiparallel. Now new strands are always formed fifth to third direction. Maturation of Okazaki fragments. DNA synthesis of lagging template takes place in the form of small fragments called Okazaki fragments. These fragments are joined by enzymes DNA ligase. Next step, number 7, formation of daughter DNA molecules. At the end of replication, two daughter DNA molecules are formed. In each daughter DNA, one strand is parental and other one is totally newly synthesized. Thus, 50% is contributed by mother DNA. Hence, it is described as semi-conservative replication. Next topic, the chapter, that is protein synthesis. Proteins are very important biomolecules. They serve as structural components, enzymes and hormones. The cell needs to synthesize new protein molecules. The process of protein synthesis includes transcription and translation. The process of copying the genetic information from one strand of DNA into a single stranded RNA transcript is termed as transcription. During this process, synthesis of complementary strand of RNA takes place. Central dogma in this concept, double-stranded DNA molecules gives rise to messenger RNA, which act as messenger to program the synthesis of a polypeptide chain. This type of unidirectional flow of information from DNA to RNA to proteins and proteins is referred as central dogma of molecular biology. It was postulated by F.H.C. Crick in 1958. They shown as figure. The presence concept of central dogma in retrovirus or riboviruses is given by Tamim in 1970 and Baltimore in 1970 as shown in the figure. It consists of number 1, transcription. During transcription, information of only one strand of DNA is copied into RNA. This strand of DNA acts as template. Enzyme RNA polymerase catalyzed by the formation of RNA transcript. DNA is located in the nucleotide of prokaryotes and in nucleus of eukaryotes. DNA transcript takes place in nucleus in eukaryotes, whereas translation occurs in cytoplasm. DNA transferred information of to messenger RNA, which then moves to ribosomes. Transcription occur in the nucleus during G1 and G2 phase of cell cycle. DNA has promoter and terminator sites. Transcription start at promoters site and stop at terminator site. Actually, the process of transcription in both prokaryotes and eukaryotes involves 3 stages: initiation, elongation and termination. Each transcription unit segment of DNA is called as transcription unit. It consists of promoter, structural gene and terminator. 2 strands of DNA in the structural gene shows following features. The promoter is located towards fifth end of structural gene, that is upstream. It is the DNA sequence that provided bind site for the enzyme RNA polymerase. RNA polymerase bind to specific promoter In prokaryotes, the enzyme recognizes the promoter by its sigma factors subunits. Polymerase catalyzed polymerization in fifth to third direction. So the DNA strand having third carbon to fifth carbon polarity act as template strand. Next, the terminator is located at third end of coding strand, that is downstream. It defines the end of the transcription process as shown in the figure. After binding the promoter, RNA polymerase moves along the DNA and causes local unwinding of DNA duplex into two chains in the region of the gene exposed. ATCG bases project in the nucleoplasm as shown in the figures. Next topic or next point, that is transcription units and the genes. The DNA sequence coding for the messenger RNA oblique transfer RNA oblique ribosomal RNA is defined as a gene. Histone is a segment of DNA coding for polypeptide. A single structure gene in transcription unit is said to be monocystronic, whereas a long segment, the DNA having set of various structural genes in the transcription unit, is referred as polycystronic. Structural genes in eukaryotes have interrupted non-coding sequences. The coding sequences or express sequences are defined are exons. Only exons appear in processed messenger RNA in eukaryotes. Processing of hnRNA. In eukaryotes, forms of RNA transcribed from DNA are called as primary transcript. Such transcript undergoes changes called processing or maturation before becoming functional. Primary transcript is non-functional and contain both exons and introns. During processing, only introns are removed by the processes called as splitting as shown in the figure. Next topic, that is genetic code. It is already known that DNA is a master molecule of cell that initiates, guides, regulates and controls the process of protein synthesis. To perform the complicated functions, it must carry the requisite information for the synthesis of proteins. Obviously, this information has to be verily located in the DNA itself. The site for storing this information lies in the sequence of nucleotides, as evidenced by Yunovsky and Sarabhai in 1964. About 20 different types of amino acids are involved in the process of synthesis of protein. DNA molecules has 4 types of nitrogen bases. To identify these 20 types of amino acid, questions arises then, how is it possible that 20 types of amino acids are encoded for 4 types of nitrogen bases? According to FSC Crick, this information is stored in the form of coded language called a genetic code that contain code word codons, each one specifying specific amino acid. Genetic code, therefore, it is collection of base sequences that correspond to each amino acids. A single nitrogen base in a codon will encode of only 4 different types of amino acid. A combination of 2 nitrogen bases will specify only 16 different types of amino acid. That will be sufficient to specify 20 different types of amino acids. Thus, there would be 64 different codons in the dictionary of genetic code, and that each code word has to be triplet codon. Every 3 consecutive nucleotides in DNA will constitute a triplet codon. genetic code is a triplet codon was evidenced first by Crick in 1961 using frame shift mutation. However, M. Nirenberg and Mathei were able to synthesize artificial messenger RNA which contain only one type of nitrogenous base, that is uracil. This synthetic poly-U sequence was transferred to protein synthesizing enzymes. A small polypeptide molecules was produced by the linking of phenylalanine molecules. This explains that triple U code for phenylalanine, later different homopolymer codons were deciphered codons formed by two or more bases were also tried. Dr. Hargovind Khorana, he devised a technique for artificially synthesis of messenger RNA with repeated the sequence of known nucleotides by using synthetic DNA. Dr. Khorana prepared chains of polyribonucleotides with known repeated sequence of 2 or 3 nucleotides. Example is CUC, UCU, CUC, UCU. Next topic, that is characteristics of genetic code. Genetic code shows 10 different characteristics. Number 1, genetic code is a triplet code. The sequence of 3 consecutive bases constitute codon which specifies one particular amino acids. Base sequence in a codon is always fifth to third direction. In every living organism, genetic code is a triplet code. Number 2, genetic code has distinct polarity. It shows definite polarity, that is direction, if it therefore is always read in fifth carbon to third carbon direction and not in third carbon to fifth carbon direction. Otherwise, message will change. Example is fifth carbon AUG to third carbon. Number 3, genetic code is non-overlapping. Code is non-overlapping, that is each single base is a part of only one codon. Adjacent codon do not overlap. If non-overlapping, then with 6 consecutive bases, only 2 amino acids molecules will be in the chain. Number 4, genetic code is commaless. There is no gap mark between successive or consecutive codon. Number 5, genetic code has degeneracy. Usually single amino acid is encoded by single codon. However, some amino acids are encoded by the more than one codon. Number 6, genetic code is universal by and large in all living organisms. The specific codon specifies same amino acids. Example is codon AUG always specifies amino acid methionine in all organisms from bacteria up to humans. Number 7, genetic code is non-ambiguous. Specific amino acid is encoded by the particular codon. Alternatively, two different amino acids will never be encoded by the same codon. Number 8, initiation codon and terminated codon. AUG is always an initiation codon in any and every messenger RNA. AUG code for the amino acid methionine. Out of 64 codon, 3 codons, that is UAA, UAG and UGA, are terminated codon. Number 9, universal. Usually in all living organisms, the specific codon specifies same amino acids. And last one, 10, codon and anticodon. Codon is part of DNA. Example, AUG is codon. It is always represented as fifth carbon AUG, third carbon, anticodon is the part of transfer RNA. It is always represented as third carbon, UAC to fifth carbon.